Let us discuss how this macromolecular DNA is packed into structures known as chromosomes, the 6 feet long DNA. The DNA is packed in a unit which is known as nucleosome. 6 histone proteins are found in the core of the nucleosome and DNA coils around it. This double helical DNA coils around 6 histone proteins and this unit is known as nucleosomes, means coiled DNA around 6 histone protein units, and then this nucleosomes, they just add on and coil and supercoil, and then they form a fiber which is also known as chromatin fiber. So, the chromatin fiber is 30 nanometer in diameter. So, from 2 nanometer DNA, we have a 30 nanometer chromatin fiber which again coils and condenses and then forms the chromatin material which arranges itself in the form of chromosome structures, as you can see. So, see, it is highly now condensed and it is also supercoiled. This condensation and supercoiling has resulted in the formation of this structure which we call as chromosomes, which are visible in the prophase stage of cell division. Then eventually when these chromosomes are visible, they further go through many divisional changes, anaphase, go through telophase, and finally after the cell division there is karyokinesis. I told you that the nucleus is going to divide and after that cytokinesis, means the cytoplasm will divide, eventually resulting into the formation of 2 daughter cells. This is how this 6 feet long DNA macromolecule is packed into chromosomes which are then present in the nucleus of the cell and they are visible in the prophase stage, and then eventually they replicate and changes go on and then finally 2 daughter cells are formed.